Hello, everybody. Nice to see you. Welcome to season 15 of SoundWaves. Welcome back to those of you who were here a alarmingly short time ago. And welcome to, are any of you new to SoundWaves? Oh, great. Well, I hope you enjoy it. We've had such an amazing time over the years collaborating with now almost 60 departments and learning so much stuff that makes our world a bigger place and a more interesting place than we ever could have imagined. And also learning about music of various kinds. And this is our first time that we'll be having Indian music. And I'll be playing Indian music for the first time. And so that'll be pretty exciting. So this is our new format that we're doing, where we're starting with a little prelude music. And then we're going to have 3 talks, and then a music talk, and then the main portion of the music, which will be longer than what you're used to if you've been coming for many years. So I think we will get right into it, and I'll welcome Samita to the stage.
जमुना किनारे मोरा गाँव जमुना किनारे मोरा गाँव हाँ सावरे ऐ चैयो जमुना किनारे मोरी ऊंची हवेली जमुना किनारे मोरी ऊंची हवेली मैं ब्रिज की गोपिका नवेली राधा रंगीली मेरो नाम राधा रंगीली मेरो नाम हाँ सावरे ऐ जयो सावरे ऐ जयो सावरे ऐ जयो सावरे ऐ जयो सुन मुरली में भई रे दीवानी सुन मुरली में भई रे दीवानी सुध बुध खोई नींद गवाई सुध बुध खोई नींद गवाई ध्यान लग्यो है तेरो नाम ध्यान लग्यो है तेरो नाम हाँ सावरे ऐ जयो सावरे ऐ जयो सावरे ऐ जयो सावरे ऐ जयो ऐ जयो सावरे ऐ जयो ऐ जयो ऐ जयो ऐ जयो सावरे ऐ जयो 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 ऐ जयो
All right. Good evening, everybody. My name is Gavin Dainert. I am an emerging contaminant scientist with Wisconsin Sea Grant here on campus. And today I'm going to give a little bit of talk about PFAS in our water. and a connection between contaminants, the environment, and wildlife health. So what are PFAS, right? So PFAS are per- and polyfluoroalkyl substances, and they're a family of man-made chemicals that contain carbon, fluorine, and other elements. And they have been nicknamed what we call forever chemicals. And part of the reason they've got that nickname is they last in our environment for very, very long times. Kind of almost think about it for lasting forever. And what's really interesting about them is they are very, very small. They're about 1 to 2 micrometers in length. And to put that into comparison for you, the diameter of your human hair is about 20 micrometers, so almost 20 times smaller than that. A red blood cell that you have in your body is about 5 micrometers, so 5 times smaller than that. And the spiderweb silk is about 2 micrometers in diameter, so about half of that size as well. So you can think about them as they're very, very small. And PFAS, as I said, they're a family of chemicals, and there's about 14,000 different types. And maybe what's the biggest issue about PFAS is, as scientists, we can't even agree on how many there are. Some of us think there's a little bit over 1 million. Some of us say there's only about 4,000. But the general consensus is there's about 14,000 different types of PFAS. And 2 of the most famously studied PFAS that I have listed up here are known as PFOA and PFOS. And I'm sorry, I'm going to try my best not to do a lot of acronyms, but there's a lot of PFAS that are out there. These PFAS were actually created in the 1950s, and since then, in about 2015, both of these 2 PFAS were actually voluntarily banned due to some potential toxicity reasons. And part of the reason that they were banned is this idea is they are extremely persistent, or they're very prevalent and they last a long time. And they have half-lives that are up to 5 to 10 years. So those original PFAS that were actually made back in the 1950s, those PFAS are still in existence in our environment right now. But since they got banned, they didn't stop making different PFAS. They actually started making different types of PFAS. And they made these ones, in theory, that would degrade faster. So I'll point out in there, you can see these oxygens, or those O's that are in there, and those are actually thought of to help them degrade a lot faster. In theory, these ones can degrade potentially within 2 years, 3 years, so still not a whole lot faster, but they do degrade a little bit faster. So there's a lot of them, right? They last a long time, but what are they and why do we use them? So PFAS are in everything. They're in everything that can be oil resistant. They're in things that are water resistant, heat resistant. Basically anything that can be resistant of something, PFAS are probably in them. And a lot of us right now are probably either one, wearing something that has PFAS in them, or we've dealt with PFAS today. The carpet your feet are sitting on definitely have PFAS in them. And so PFAS are mainly most notable in 2 different things: aqueous firefighting foams and nonstick coating pans, so all of your Teflon pans that you use. But they're also, like I said, in a lot of other things, like your paper products. They're in architectural resins. They can be even in your McDonald's wrapper that some of us probably had for breakfast this morning. So they're in a lot of different things. And maybe the most important to think about them is PFAS is a global contaminant. So I know we're talking about the micro scale a little bit today, but this is a macro problem. This is a global problem. And I put this figure up here just to kind of give you an idea of 2 different things. Those 2 PFAS, PFOA and PFOS that I talked about at the beginning, those are known as the legacy PFAS. And as you can see, in all of those green portions of that pie chart there, those are the levels of PFAS or proportions of PFAS that are found across the globe. So they're found in North America, South America, Asia, Africa. They're found in Antarctica. They're found on the top of Mount Everest. So they are found really everywhere. But I think what's the most important about this graph that I'm showing here is that red portion. And all of those are those next generation PFAS. So these studies are looking at, they're about 2020 to 2022, and you can see that the majority of PFAS that we're actually measuring are actually those next generation ones. So the old ones are still existing, right? They're not breaking down anytime soon, but we're overloading it with these new PFAS. And when we zoom in a little bit, right, to the United States, there's also like a clear unequal distribution of PFAS that's known in the United States. And I just kind of show this graph is so these blue dots, the light blue dots, these are all the PFAS levels that have been detected. So at kind of low levels, and you can see there's a lot of them. Those dark blue dots, these are PFAS levels that are above what we'll call health standards. And then the purple dots that you see there, these are all associated with military sites, or these are going to be these areas where these PFAS are very, very high levels. And what I like to kind of notice, we're in Wisconsin here. Can anybody see Wisconsin on this map? No, it's pretty hard. So like all of the Midwest, you know, kind of Minnesota, Wisconsin, Michigan, we have a lot of PFAS detection. I'd like to say we can thank 3M a little bit for that. But part of the reason that we have so many is we actually are doing a really good job of sampling it, going out and detecting it, which I'll talk a little bit about. So these areas in like Montana, Idaho, et cetera, they just don't do as much sampling. So likely there's a lot of sites out there that also have these PFAS levels. And as of August of 2025, so a couple months ago now, there's over 9,500 different sites that have been known to have elevated levels of PFAS. And that number is just going to continue to increase as we do a lot more sampling. So again, it's all over the place, right? It lasts a very long time, but why should we care about PFAS, right? PFAS can be toxic to animals as well as humans. And what we know is that from epidemiology studies or studies on humans as well as lab rodents, that these PFAS can have negative effects. In particular, studies have found evidence that high levels of PFAS can increase your cholesterol, they can decrease fertility in women, and they can lead to a decreased antibody response. Or in theory, you can think about that as like weakening your immune system when you're trying to deal with diseases. And a lot of these studies that have been done in humans have actually shown that these very high levels of PFAS exposure have reduced your response to things like vaccinations and vaccine boosters, which is obviously very prevalent, right, when we're thinking about things like COVID that, you know, have been happening in the last couple of years. But PFAS is also linked with a whole bunch of other diseases. PFAS have been thought to have impacts on thyroid disease, different types of cancer, different types of cancers, so yeah, lots of different types of diseases that can be. And some people think about this as like, oh, that doesn't make a lot of sense. It's one family of chemicals. But because there's so many different types of PFAS out there, they're all going to be working on different parts of our body. And that's why we see so many different diseases that are connected to them. So what do we know about PFAS in our environment? And I say this kind of tongue-in-cheek because I've been showing this a little bit, that there's a lot of them out there, but we're really at our infancy stage of understanding PFAS in our world and really what does it do. So since there's very little information that's out there, what we start with usually is looking at water samples, soil samples, and even air samples to kind of start to answer some thought to be some pretty basic questions about where are PFAS in our environment? What concentrations of PFAS do we see? Like what areas are going to be very high? And then maybe in particular, what types of PFAS are we actually finding in different areas? I had mentioned at the beginning that there's 14,000 of these out there, but our ability to measure them, we can only actually look at 40 of them in when we do these sample analysis. So even all the kind of things that I'm going to talk about, we really only know maybe a very micro scale of what is potentially out there in the environment. And so through testing of these different matrices like air, soil, and water, we know that PFAS contamination is mostly a point source pollution problem. And when I mean point source, I mean a contamination that is actually being directly put into our environment. And this is really important when you think about these high levels of PFAS. But on a different scale, PFAS is a ubiquitous problem. Like I said, PFAS is everywhere. You're finding it in Mount Everest, Great Lakes, etc. But those levels of PFAS that we're finding are at much low levels. And a lot of this work that's been looking at the air, the soil, and the water have been able to help us pinpoint some of these potential sources of PFAS. What we know is that these military bases, these ones that use those firefighting foams, they are a direct source to PFAS in our environment. But other sources like factories that actually create PFAS, they're emitting it into the air, which is allowing it to move to a lot of different areas, as well as landfills, right? So like I said, we're wearing a lot of it, we're throwing a lot of that garbage away, it's then getting into our landfills and then leaching into our environments. And a lot of what's really interesting about these PFAS is, like I was kind of mentioning, there's so many of them. Some of them are small, some of them are a little bit larger, and these small chain ones, let's say they're 4 or 5 chain lengths long, they're the ones that are really going to be transported through our air, and they move a lot farther. So you can find these even in rural Wisconsin in an area that has, well, thought to be pristine. But some of these larger PFAS, like PFOA and PFOS, they will concentrate very high near a pollution site. So they're really good at helping us understand who's the polluter, or how do we potentially identify the cause of PFAS contamination in our environment. And what's really cool about looking at water compared to sediment is the levels of PFAS that are going to be in the water are going to be usually those short chain ones, but then when you get to the sediment levels, you're going to see a lot of those longer chain ones and at much higher concentrations. And then when we dive into Wisconsin, I know we're all from Wisconsin here, so I like to always point this out. We do a couple different ways to kind of look at PFAS. We have the graph on your all's left. These are long-term sites. So these are going to be sites that have been looking at PFAS for 10 to 20 different years. So we can look at this in a way of how are these levels changing? What types of PFAS are we finding? Are we seeing new ones? Are we seeing the old ones still exist? But then on the right, your right, you're going to be seeing these are all the PFAS samples that are usually done to protect human health. So a lot of these are looking at PFAS in drinking water or public sources, whether it be public drinking water or private wells. And you can tell there's a lot of different PFAS levels that are being found. So what we know, obviously, as I'm kind of preaching a little bit, is like groundwater, drinking water, soil, and air, they can all be contaminated by PFAS. So while it's a micro problem, as in the sense they're very small, it is all over the place. But this whole theme is talking about scaling up. So how does PFAS potentially scale up? How do these microcontaminants move from the environment into, let's say, higher organisms? And I'm going to kind of try to talk about this in 3 different ways. How does it get into our plants? How does it get into our fish? And then how does it get into top predators, let's say like bald eagles? So first, some of the work that we do in our lab is looking at how does this PFAS move from the environment into different plant species. And particularly, we've been looking at maple sap and maple syrup. So what we've actually gone out there is asked these questions of like, do trees even take up PFAS? Is it a good way for us to understand PFAS contamination in the environment? So I really like to think about this as like, what can the trees tell us? Are the trees going to be a good indicator for us to understand contamination in an area? In one specific way, like I said, is we will go out to a sugar bush. You guys have maybe seen some of these blue bags that are sitting out there that's collecting maple sap in the spring. And we can go to these vast areas and we can actually collect the sap samples and we can measure the PFAS levels in that. And what we've been able to find is as you go to these areas that, let's say they're a mile long or a mile in radius, they give us a really good snapshot of what levels of PFAS are there in the environment there. And then they do a good job of letting us know, is the PFAS coming from the water? Is it coming from the soil that the trees are living in? Or is it coming from the air and that they're actually able to take it in through the leaves? We've been finding basically that you can have PFAS levels in these maple sap, but maybe what's the most important is when you take the syrup of the sap, you boil down the sap about 40 times, you can actually find a lot more PFAS. You find it at a lot more higher concentrations, which is a little scary for somebody like me that loves to eat maple sap. But the great news, usually when I'm a PFAS talker, I give you bad news. The levels of PFAS that we're finding in your sap or syrup are below health concerns. So we can all take a break there. But what we're really trying to try to do now is like I said, we're at that infancy stage. Where does the PFAS go in them? Does it go in the tree or the leaves? Does it go in the bark? Does it go into the different parts of the tree? So we know it can get into the environment. We know it gets into the plants and the trees. But how does PFAS potentially accumulate or bioaccumulate from our water into different fish or different other animals? And really what we want to know is how does this contaminant move throughout the water and then into the animals, and where does it go? So then I ask you the question, so what can the fish really tell us? So in our work, what we've been doing is measuring a lot of fish fillets for PFAS levels to really understand what are the levels in the water and then what are the levels in the fish. We know that PFAS can move through a couple different ways. One, it can be absorbed through the skin. Two, it can be absorbed while they're eating other animals or digesting other food sources, but it can also cross the gills into their bloodstream. So it can get into their body a lot of different ways. But maybe what's the most fascinating for me is where does it go once it's in the body? Maybe the obvious one is because it's a contaminant, it will get to high levels in your liver, or in the fish's liver, sorry, ours too. But maybe what's a little different is it can actually cross that blood-brain barrier. So we see very, very high levels of this PFAS in our brains, or in the fish brains, sorry. But maybe the saddest part is PFAS will actually move into the fillet. The fillet is what we all eat, right? We've all had a Wisconsin fish fry here. So these PFAS will actually accumulate very, very high levels in our fish fillets. And that's really interesting because what it's telling us is, well, these PFAS, they could be hydrophilic, meaning they like to be in water. They can also be lipophilic, meaning they like to be in fats. But they are also, because we're finding it in the tissues, they're proteinophilic, meaning they like to aggregate to proteins, which is why we're seeing them at high levels in our fish fillets. I don't know how many of us are out fishing, but just as a fair warning, we do actually have a PFOS fish consumption guidelines for even Lake Monona in our backyard here. So we're actually recommended to eat about 1 fish a month in that, out of that system. So PFAS gets into our fish, right? Gets into our plants. But another question I'm going the wrong direction, sorry. The next question we might ask is how does it magnify? And what I mean by that is how does it go from one trophic level to the next, all the way up to top predators? And when we think about top predators, right, we can think about things like wolves on the terrestrial environment. We can think about humans. We're a top predator. We eat a lot of different things, but also like bald eagles. So what can bald eagles tell us about PFAS? So we, as our lab group, we assess PFAS in baby bald eagles for 2 different reasons. 1, what are their levels? But 2, also, what are the health impacts that are on them? And what we've learned is that PFAS can actually get into our bald eagles at levels that are almost 1 part per million, which is about 100 to 1,000 times higher than what you're seeing in the fish, and almost 100,000 times higher than what you're seeing in the water. So it does seem to biomagnify or get to higher levels in these trophics. And then what we can do with that is once we know what their levels are, we can actually take a step back and we can say, well, how healthy are these birds? So Going back, all right, we've talked about scaling up, right? These PFAS have moved up all the way up in the environment. But I'm going to try to scale us a little bit back down and ask, like, what are these PFAS doing to these birds? And in short, what we're seeing is that they can have impacts on the immune system. They can have impacts on the endocrine system, which is like your stress hormones or your reproductive hormones, et cetera. And then when you get all the way down back, I'm going to take you all the way back down to the micro scale, is that they can have impacts even on your cells that you are producing. So you have different white blood cells that are going to be promoted when you have low levels of PFAS compared to high levels of PFAS. And then even more is it can have impacts on your gene expression and particularly even on just DNA degradation. So this PFAS is a scalable issue, right? It's a very, very small thing that can have that can move all the way up in the environment, can get all the way to these top levels of animals, but then scaling back down, right, it can have these impacts on such intricate parts of our body. So PFAS pollution, right, connects the global production, regional sources, and ecological health, and it's illustrating kind of this far-reaching consequences of this industry of creating PFAS. So with that, thank you very much.
So thank you so much for coming today. My name is Karthik Anantharaman. I'm a professor in the Department of Bacteriology here at UW-Madison. And I'm going to talk to you about viruses. And again, I'll talk to you about different scales associated with viruses. Viruses are tiny, so the micro scale is very apparent, but I'll also hopefully, from my talk, you can get a sense for how big their impacts are. And to start off, let's discuss what a microbiome is. I'm not going to start off about health or ecosystems, but let's talk about microbiomes. When you think of the word microbiome, a microbiome is a collection of different kinds of organisms. What are these organisms? There is bacteria. Bacteria typically, exist everywhere in the environment. They cause a lot of diseases. We've heard about bacteria, but they also contain what are called archaea. Archaea are organisms that are very similar to bacteria. They're not as well studied, but they're literally everywhere. What is not well known about microbiomes is that there is also a lot of viruses and other kinds of organisms such as fungi. And when you think about viruses, what I want you to get out of the 20 minutes that I have today is really their abundance and their importance. Now, when we think of microbiomes, the history of microbiomes is pretty ancient. It's not as if microbiomes just came about recently. Microbiomes have been around for pretty much as long as the Earth has. The Earth, came about 4.5 billion years ago. So the sun came on about 5 billion years ago. The Earth came about 4.5 billion years ago. The first cells appeared on our planet about 4 billion years ago. And no one knows exactly when viruses came about, but the best guess, or the best guesses we have, is that when there were bacteria about 4 billion years ago, viruses should have come about. And if you're wondering where humans are, imagine 4 billion years and imagine 0.002 billion years. We are right here. So on the scale, we came about very, very recently, right? So that gives you a sense of perspective to how long bacteria have been on this planet, how long viruses have been on this planet. Now, when you think about microbiomes, microbiomes are everywhere, as I said. So there are microbiomes in the environment, and there are microbiomes associated with humans. When you think about environmental microbiomes, you can think about microbiomes pretty much anywhere. The carpet over here has bacteria, has viruses, so there is a microbiome associated with carpets over here. There's a microbiome associated with soil, with the aquatic systems. My lab studies aquatic microbiomes, and in the context of the aquatic microbiomes, today I'll talk to you a little bit about marine microbiomes. When you think about microbiomes in the oceans, there are different kinds. Ocean microbiomes are very complex. Imagine an ocean water imagine the ocean. The surface ocean is very different to the deep ocean, right? The surface ocean has sunlight coming in, there's a lot of fish, there's plankton, there's a lot of activity. The deep ocean is dark. There's not much down there. There's few animals, but there are a lot of bacteria and there are a lot of viruses. And we study these viruses to try and understand them and then sort of apply them in other contexts. What is important to know about microbiomes, and you'll see this in both environmental microbiomes as well as human microbiomes, is that they're very heterogeneous. So the microbiome of the surface ocean is very different than the microbiome of the deep ocean. The microbiome of the Pacific Ocean is going to be very different than the microbiome of the Atlantic Ocean, as an example. And if you took just two lakes here in Madison, if you looked at the microbiome of Lake Mendota, it's going to be different than the microbiome of Lake Monona. When you think about human microbiomes, we have now begun to understand that when you say the word microbiome, it's not just bacteria. For the longest time, when we sort of used the word microbiome, we pretty much only meant bacteria. But recently, we've sort of encompassed the word microbiome to also include viruses and a few other sort of organisms like fungi I mentioned. Now, what's important to understand about the microbiome is that the microbiome, no matter where it is, in the environment or in human systems, doesn't operate in a vacuum. The microbiome is constantly interacting with the host genome. So in a human context, it's interacting with our genome. And when it's interacting with the human genome, it sort of elicits a host immune response. Now, when there is a host immune response, this can sort of go in two directions. Either it can protect against certain diseases, or it can cause diseases. So either it's protecting or, say, potentiating diseases. And I'll give you some examples about both. The other aspect of viruses in the human microbiomes is that, yes, when the viruses are interacting with the host genome, they can drive diseases. But what about us? Do we have an impact on the microbiome, or specifically the viruses in the microbiome? And the answer is yes. And this happens in a variety of ways. It can happen from something as simple as diet. So the kind of diet you consume impacts the microbiome. So it impacts the viruses that are present in the microbiome. It can arise from genetics. So different people are going to have different kinds of microbiomes. It can come from disease. So, different microbiomes are associated with whether someone's healthy or if someone has a particular disease. And finally, it also, microbiomes are associated with us from very early childhood. And one of the ways that happens is as a child begins to grow and begins to consume food, imagine the different kinds of food a newborn has. Either a newborn is having breast milk or a newborn is having some sort of a formula. And depending on what a newborn gets, they're going to have a different kind of a microbiome. And that can have long-term impacts on children as well. So with this sort of a background, what I want you to get out of it is that when it comes to viruses in microbiomes, they're just very poorly studied. So that's the entire motivation of my research program. We want to study viruses in microbiomes and then eventually use them in some sort of an applied or biotechnological context. We call ourselves ecologists. Even though we are studying microbiomes, we use computational approaches, we use AI, but fundamentally we are ecologists. So when it comes to viruses, the kinds of questions my group asks are, what is the role of viruses in an ecosystem or in a microbiome? Most often, when we say viruses, we think of, let's say, the coronavirus that caused the epidemic. We think of the flu. We think of HIV. But the reality is these are not the most abundant viruses. Okay, like if you took all of the flu viruses, the viruses that infect bacteria are just going to outnumber them by orders of magnitude. So the human viruses are not as important on our planet as other kinds of viruses. In fact, a lot of the beneficial viruses are definitely going to outnumber the kinds of viruses that cause diseases. Beyond this, we are also very interested in understanding everything associated with viruses. Who are these viruses? Who are they interacting with? Are they interacting with bacteria? Are they interacting with humans? So all of this is something that we study in our lab. How do we do this? We do this by a process that's known as omics. Very broadly speaking, omics is an umbrella term, and it refers to anything and everything that can be sequenced. So the idea is that anything that has DNA or other forms of nucleic acids like RNA can be sequenced one way or another, and once it's sequenced, you can sort of derive a lot of information from it. This is how we sequence our human genome. This is how we would sequence the microbiome, and any sort of determination of about the human genome diseases is all done through omics. So in our laboratory, about 50% of what we do is associated with bioinformatics and bioengineering, and the remaining 50% is very broad microbiome research. So we study all kinds of microbiomes, human microbiomes, freshwater microbiomes. In fact, we've been studying, we have an amazing data set right here in Lake Mendota, where we've been studying it since 1999. So we have microbiomes and viruses of Lake Mendota studied since 1999 up until today. And this is the best sort of viral data set out there on the planet for at least the environment. Now, my laboratory also studies sulfur. Now, I'm not going to use a lot of acronyms or say technical terms. I'm going to try and reduce that. But we do study sulfur metabolism, and we study this in different contexts. And I'm going to try and explain this the best I can so that I can tie in environmental microbiomes to human microbiomes. In the environment, typically we are studying inorganic sulfur. Be it in the oceans or be it in freshwater, we have field sites. So me, my students, my lab members, we're all going out to the ocean. We do all of our sampling ourselves. So we build instruments, we go down in submarines, or we use robots to sample the oceans and specifically sample the viruses. And we do that with Lake Mendota as well. We take a boat out and we sample Lake Mendota. And when it comes to sampling humans, it's not as interesting. There's no field work involved, but yes, we do have a lot of cohorts, patient cohorts, where we work with as well. Now, before I get into viruses, I just wanted to show an image here. Here is Alvin. This is a US Navy submarine. This is the submarine that discovered the Titanic. And what you're looking at over here is a picture of the Alvin. You can't see me because I'm stuck right inside Alvin. And if you're wondering why there is a diver right on top, this is a submarine. And believe it or not, when you're inside a submarine, when you're launched from a ship into the water, someone comes up, literally has a phone, and calls inside to ask you if everything's okay and there's no water inside. That's literally what it is right now. So with this, I'll sort of get into a primer on viruses. So what we study mostly, as I said, are phage. Phage are viruses that infect bacteria. Now, let me sort of show you this image. And this image is not super clear, but what you're looking at over here is an image of the night sky. Now, imagine a clear night, and all you can see are stars and objects in the sky. And how many stars do we think there are in the universe? We think there's about 10 to the power 29 to 10 to the power 30 stars or objects in the sky. And if you just think about how many viruses there are on Earth, it's about 10 to the power 31. So there's more viruses on Earth than all of the stars that we think exist in the universe. So you can imagine how abundant viruses are. And what does a virus do, or specifically, how does a phage operate? What's the life cycle of a phage? Very simply, a phage lands on a cell, or a virus, lands on a cell. Once it lands on the cell, it injects its DNA. Once it injects its DNA, it then begins the process of replicating inside the cell. More viruses, or baby viruses, are produced right here, and then eventually the cell dies, the viruses sort of come out. These are the shapes of most of the viruses that infect bacteria. They all have a long sort of a stalk, a head, and then legs. This is a very standard shape of different kinds of viruses that infect bacteria. And if you run a simple example, this is something I show a lot of my students. You can run a simple example in a class. And if you just grow bacteria in a tube, you will see a very milky suspension that looks like this. And if you just introduce a few viruses into that mix, viruses slowly begin to kill the bacteria, they propagate, and eventually you end up with a very clear solution that looks like this. So if you have a clear solution, then there's very few bacteria. If you have something very cloudy, there's a lot of bacteria in there. But what my lab specifically studies, and what we are very interested in, is a process known as viral auxiliary metabolism. Again, very poorly studied process, but what is it? Now, imagine, since we are talking about the oceans, I thought I'd give you an example associated with pirates in the oceans. And AMGs, or auxiliary metabolic genes, are genes that viruses steal from their hosts, keep them on their genomes, and then over time or over evolution sort of use it to manipulate their host, to trick their hosts. Now, what's in this for the host and what's in this for the virus? Okay? So imagine this scenario. The way we think about viruses, viruses infect cells, they kill cells, that's how we think about it. So imagine a pirate. A pirate comes in, takes over a ship, they pillage and they plunder, right? So the viruses infect the cell, they replicate, they make baby viruses, and then they get out. This is how we imagine viruses actually operate. But what seems to happen in the environment is that viruses can get in, infect a cell, use these AMGs, or auxiliary metabolic genes, and then rather than killing the cell, sort of keep it alive for a little longer. So what's in it for the host? The host is being kept alive longer than it otherwise would be. And what's in it for the virus? The virus doesn't kill the cell. Over here, imagine this. There's 1, 2, 3, 4, 5 different baby viruses that were produced by one cell. But if the virus keeps the host alive for much longer, it can produce much more baby viruses over time. So that's the idea here behind using these AMGs. Now let me show you a video of deep ocean viruses or viruses in the oceans, and then I'll sort of tie it in to human diseases.
I want people to associate viruses with the health of the oceans. Where there is life, there are viruses, and we can't see them, but sure enough, they're there, and the health of all of those animals, of all of those rich colors of biofilms really depends on the viruses. They are more abundant than all other biology by orders of magnitude. Much like large predators, viruses are just turning over the ecosystem and keeping things in check. Much of the research is really focused on viruses present in the surface oceans. It's microorganisms in the dark oceans critical to keeping oceans healthy, and it's these viruses that we know next to nothing about and that I'm here to study. I think hydrothermal plumes likely host one of the most diverse ecosystems, primarily coming from the presence of so many different chemicals in there. Back in 2014, we were part of a project that we discovered viruses that can actually manipulate bacterial hosts, and the way they do it is that the bacteria store sulfur inside their bodies as a food reserve. What the viruses would do is trick the bacteria into using that sulfur, and the bacteria keeps burning that sulfur, but the viruses derive all of that energy. And this is a very, very unique form of metabolism that's been observed nowhere other than in hydrothermal vents. We have now taken a lot of these findings and are applying them to our understanding of human diseases, especially the human gastrointestinal tract that are associated with sulfur. And that would not have happened if we had not been down there studying hydrothermal vent viruses. I think those mirror pools are a hotbed of viral activity.
What's that underneath liquid? Well,
it's hydrothermal fluid. It's primordial juice, fueling all of the biology that you see around the system.
The viruses we were going to find in here, we're not going to find anywhere else. Really, really unique, and I think it's really important to capture their diversity and understand their function. Think about the vastness of the ocean right in front of us. And all of this while, when we have been studying viruses, we have only been filtering small amounts of water. And that has its own sense of biases that come along with it. To think of, say, traditional virus filtration methods, it's about 5 milliliters a minute. So now you can see water coming in. Water is coming in through this pipe that's connected to these CTDs outside. So this is an inline filtration system. So what you're capturing over here are the largest microbes. The last filter is viruses. And you can see how quickly it's going. This is designed to go somewhere around 1,000 times quicker than a traditional filter. So we can use these samples for genomics, metabolomics, all of this from just one set of samples. The first time that anyone's been able to achieve it at this magnitude, and we are immensely proud of this. The fact that we will be able to study microorganisms and viruses in their natural habitat here will help us learn about how they might respond to future global change. Sometimes the most powerful things cannot be seen, and I tend to associate viruses with that.
Oops. So if you saw the video there, like we don't study any of the octopuses or the lobsters or any of those animals, but they make for beautiful imagery. So we do include them from time to time. But just taking a lot of the cues that we've observed in the ocean, studying volcanoes, studying hydrothermal vents, what can we take away from there? And one of the things that we took away is that when it comes to the human gut, very little is known about viruses and the processes associated with these AMGs. So one of the first questions we asked was if these AMGs are actually present in the human gut. So we investigated human microbiomes and wanted to know if these AMGs are actually present. And it turns out that when you look at different microbiomes, AMGs are very abundant, and it's always different AMGs because they're associated with different microbiomes. So as an example, our oral microbiome, what's inside our mouth, is very different than what's inside our gut. So you see different microbiomes are associated with different viruses and different AMGs. But the most important takeaway is that a lot of them seem to be associated with this particular molecule known as H2S, or hydrogen sulfide. Hydrogen sulfide is a highly toxic compound, and it's a known risk factor for gastrointestinal disorders. So if you think about H2S production, it's a well-known sort of risk factor for a lot of gastrointestinal disorders such as Crohn's disease, IBD or intestinal bowel disease, colon cancer, colorectal cancer. How does this happen? If you think about a healthy human gut has very little H2S. You have a colonic epithelial lining over here, and you have a mucus lining. All of this is intact. But if you have H2S present at very high concentrations, all of a sudden the mucus lining breaks down, viruses invade, and there's a lot of inflammation. And where is this H2S actually coming from? A lot of this is actually coming from taurine, from sulfate, and from amino acids, cysteine and methionine. So some of these are produced in the body, but a lot of them are actually associated with what we call the Western diet. What is the Western diet? The Western diet is an animal-rich, protein-heavy diet that's very low on fibers. So if your doctor or your dietitian ever tells you, consume more fibers, or we tell children, I certainly tell my kid, he doesn't listen, about eating his vegetables, it's for this reason. You have to balance your meats with your vegetables, and eating vegetables actually, or fiber, keeps your H2S concentrations down, and that's good for your gut. How does this happen? How do viruses actually produce H2S, and how do they impact our gut? Imagine this scenario. There are viruses in your gut that have genes to degrade a specific compound, cysteine. And when the viruses have that, they can actually cause uninhibited cysteine degradation, which sort of produces H2S. So the way this works is every bacterial cell in the human body has a lot of cysteine, which is an amino acid that contains sulfur. Let's say a phage lands on this particular bacterium right here. As soon as the phage lands on the bacterium, it begins its infection process, and it expresses these AMGs. The bacteria are being forced to upregulate their cysteine degradation and produce the H2S, and eventually viruses replicate inside the host cell, which releases H2S, and that causes more intestinal inflammation. So if you think about how this actually impacts the gut, if you had a healthy gut, you should not see any inflammation. If you had only bacteria producing H2S, you will see some inflammation. But if you had both bacteria and viruses driving H2S production, you're going to see far greater amounts of intestinal inflammation, and this is what drives disease. So to end, if there's one thing I want you all to take away from here, just think of microbiomes as being everywhere. If you think about your skin, your gut, microbiomes are everywhere. There's lots of bacteria, there's lots of viruses, and we don't need to be scared of them. At the end of the day, we should see microbiomes as a natural resource. We should also see viruses as a natural resource, and we can harness them for mitigating impacts such as climate change or even for treatment of specific diseases. So with that, thank you so much.
Good evening. My name is David Johnson. I'm a senior lecturer in the Department of Economics here at our beloved UW-Madison. If I hear the word scale, I immediately think of tariffs. And I want to tell you a story that's not true. Right after 7:30 this morning, when I sent Dan these slides, here is the story that is not true, but sounds really good. I emailed these slides to my colleague, Kim Rule, who currently serves on President Trump's Council of Economic Advisors. And I said to Kim, I hope things are going well. If you have a chance today, run them by the president. I think they might be persuasive. This afternoon, President Trump signed an executive order scaling back a significant number of tariffs on food products. So, what I want to try to motivate for you today is that there is a micro of trade and a macro of trade. And when we concentrate on the micro of trade, we miss the macro impacts, the very macro impacts that I think were behind the president's executive order today. But first, a little bit of the difference between being a microeconomist and a macroeconomist. So microeconomists study how households and firms make decisions and how they interact in markets, whereas macroeconomists study economy-wide phenomena, including inflation, unemployment, and economic growth. Now, this sounds very textbooky. What's really the difference between a microeconomist and a macroeconomist? One letter. Also, macroeconomists are much less self-centered than microeconomists because I has been replaced by A. Or potentially my favorite difference is that microeconomists are wrong about small things, whereas macroeconomists are wrong about things in general. But if I'm trying to give you the motivation for why we continue to have presidents of the United States who believe in tariffs, I have to go back and tell you a little story. What is a tariff? A tariff is nothing more than a tax on imports. What are imports? Imports are goods that are produced abroad and consumed domestically. And the reason this is one of my favorite examples to instruct you on the micro and macro of trade and tariffs is that it targeted a specific industry. This is President Barack Obama's 2012 State of the Union address. Over 1,000 Americans are working today because we stopped a surge in Chinese tires. Meaning what? The president, Barack Obama, put tariffs on goods coming from China, specifically tires coming from China. We, as macroeconomists, look at the macro impact of what seems like a very targeted policy. I am potentially protecting a single industry in the United States. That would be domestic or American tire manufacturers. According to analysis by the Peterson Institute for International Economics, why do I pick the Peterson Institute? It is the gold standard for analyzing anything to do with international economics, international trade, international finance. It is, I am going to say this word, you're going to think I'm lying, it is nonpartisan. Okay? And they did a very deep dive, since we've been talking about oceans, into Barack Obama's Chinese tire tariffs. And they concluded that the tariffs on Chinese tires saved at most 1,200 jobs. My question to you seems very simple, but very difficult to ferret out. At what cost? The total cost to American consumers from higher prices resulting from safeguard tariffs on Chinese tires was around $1.1 billion in 2011. They were slapped on in 2009. The first year they really bit was 2011. The cost per manufacturing job saved was at least $900,000 in that year. I teach a lot of young people at this institution. I teach a lot of first and second year students at this institution. And I love thought experiments like saying the following: had we cut each of those workers a check for $800,000 and pushed them into early retirement, we would have been out ahead. The micro of trade. I'm going to target what I believe, if I'm President Obama, and don't worry, I'll get to the other presidents as well. I think I'm targeting those workers in domestic U.S. tire manufacturing. How many workers total are in domestic tire manufacturing versus how many consumers in the United States buy tires? But wait, there's more. Everything has an opportunity cost, including, or especially, an extra $1.1 billion spent on tires. Where does that money come from? That may be restaurant money. It might be latte money. It might be vacation money. It might be apparel and holiday shopping money. Doesn't that factor into the equation? Oh my, it does. Tire protectionism cost the United States economy around 2,531 jobs, when losses in the retail sector are offset against those 1,200 jobs gained or saved in tire manufacturing. Now, I went back to that State of the Union address from President Obama, and this phrase was not in it. 3,000 more people are not working in the retail sector because we stopped a surge in Chinese tires. But that is a real result, and that's what results from concentrating on the micro of trade and ignoring the macro of trade. Wow. Okay, I told you I teach a lot of first and second year students. When I talk to them about my favorite example of the tire tariffs that Barack Obama put on imports from China, I normally get two responses. Who? Then the second response, dude, I was 6. Okay? So I'm going to ask you, what about 2025? Let's take the presidents from Barack Obama to Trump 2. Barack Obama, tariffs on Chinese tires. President Trump 1, tariffs on Chinese steel and aluminum. President Joe Biden, no change in President Trump 1's tariffs on Chinese steel and aluminum. He kept them in, and for good measure, President Biden added tariffs on what? On solar panels and electric vehicles. And then we come to President Trump 2, which has put tariffs, who has put tariffs on pretty much everything under the sun. And right now, because even today, this is an enormous state of flux, the Peterson Institute also running the numbers, saying the prices paid to foreign sellers for many imported consumer goods have changed very little. Now, you may not have a degree in economics, but how do I translate that into plain English? The plain English is that those tariffs are being paid almost 100% by American consumers and American businesses. A couple of President Trump's recent posts on trade policy. I will tell you everything that I know about what it's like to teach in this current environment. It is extraordinarily challenging. I could do well not to mention politics in any lecture of Econ 101 or Econ 102. I'm not going to do that. 3 days after President Trump took the oath of office for the second time, the spring semester of 2025 began at UW-Madison. Every single lecture I give starts with 15 minutes of current events. And because Trump had been inaugurated 3 days before that first lecture, the first thing I wrote on the board for 300 people to see was President Trump, Tariff Man. These are issues we need to be discussing with the young people of this university. When we decide that we're not going to talk about politics, we're not going to talk about deep macroeconomic issues, I believe we insult the collective intelligence of the students in front of us. So, I'm going to show you what President Trump recently tweeted, whatever the social media term is now. People who oppose tariffs are fools. Well, my friends, count me as a fool. Count 98% of the people in my profession as a fool. And my tariffs are bringing in hundreds of billions of dollars. Yes, hundreds of billions of dollars out of the pockets of American consumers and out of the coffers of American businesses. And then I thought, well, why stop there? People who oppose the federal income tax, therefore, must be fools as well. Last year, the federal government collected $2.4 trillion in personal income taxes. That doesn't seem to be something that the president is talking about. Which means what? I want to leave you with the following. When we concentrate on the micro of trade, we miss the macro impact. It's easy for us to say, hey, I want to help some of those people in tire manufacturing who cannot compete with foreign imports. But if I were to tell you that we're paying $900,000 for each one of those jobs saved, I think you might raise your hand and say, maybe I'm going to reconsider. Many, many trade economists love to talk about reasons that American growth, that US economic growth, has been so incredibly vibrant for centuries. I can give you a very, very good reason. Because we in the United States live in the greatest free trade zone the world has ever known. It's called the 50 states. And so we take for granted, essentially, what the European common market has been trying to do since World War II, to get rid of barriers to trade, to go to a common currency, and try to get what we have enjoyed, maybe without really knowing it, since the birth of the country. Last, this is a little out there, but I'm a little out there too. This is a very, very famous quote, of course, from FDR's first inaugural address. I have a slight tweak that I'd like to share with you. The only thing we really have to fear is ignorance, because ignorance begets fear. And the only solution to ignorance is education at scale. Thank you very much.
Wow, thanks to all three of you. Fantastic, great lectures. Now we're at the portion of the program where we're talking about music. We don't have any visuals, so we can get rid of our screen, and we're just going to listen and try to learn by ear. So we are playing Indian music. You already heard a little bit of that at the beginning of the program. And this music is different. It's constructed differently from Western music. It uses different scales. Basically, a scale is a way of dividing up an octave, two notes that sound pretty much the same. You can hear that those notes are basically the same name of note. And we divide the octave up into 12 different parts. And in Western music, we take those parts in order. That's called a chromatic scale. And we have major scales. And we have minor scales. I'll just quickly play a cool one. Indian music divides up that span. Its micro-division is a little bit different, and the whole concept of it is different. So we're talking about ragas here. Tell us about how Indian music approaches the division of the scale.
Sure. So good evening, everyone. It's such a pleasure to be here as part of this event. And yeah, Indian music. So I've studied the classical music from North India, which is Hindustani music. There's two major traditions of classical music that come from India. From the south, there's Carnatic music, and from the north, there's Hindustani music. And the scales are shared by both. They are a little different between the two traditions, but many of them are shared. And just as Dan explained, a scale or a raga in Indian music is made up of notes. But some of the differences are that the notes are thought to be not separate from each other. So they're connected. The way that you sing or play the notes in a raga, it's how you present the notes in relation to each other. And people who have studied Hindustani classical music, if they hear that an artist is going to present a particular raga, they have certain expectations of the phrases that they'll hear, the emphasis on the notes that they're going to hear in that presentation of the scale, and maybe also the emotion that it might bring about. So I can maybe demonstrate a little of what I'm talking about.
Please do. While we get this set up, I'll point out that it's a really interesting mix of the ancient and the modern, because we have this very, very ancient tradition of singing. And we have a drone produced by an app and a synthesizer that was made last year. And we're sitting on the floor on the Iranian carpet that's old. So we're really mixing old and new. But the idea of the ragas is very old.
It is. These are ancient ragas, scales that have been sung for generations and generations passed through oral tradition. And just to add to the elements of all of the things that we have in the mix here, a Canadian-born girl of Indian descent. So my parents are first-generation immigrants from India, but I was born in Canada and raised there and learned this music in Canada, but then had the opportunity to travel and study in India from teachers there as well.
Meets a klezmer French horn player,
Yeah.
Jewish, playing a lot of music from Eastern Europe, and now playing this instrument as well. And I play bass lines on this when I play klezmer.
Right.
But I'm going to play Indian music on it, or try today. So can you sing a little bit of a raga so we can hear the pattern of notes and how they flow together?
Definitely. So the piece that we're going to present in a little bit after we're done our chat is in a rag called Hamsadwani. And this is actually a rag that has come into Hindustani music from Carnatic music. It's a pentatonic major scale. So in Indian music, the solfege, do, re, mi, fa, sol, la, ti, do, it's all the same notes, but it's sa, re, ga, ma, pa, dha, ni, sa, ni, dha, pa, ma, ga, re, sa. And this scale is sa, re, ga, pa, ni, sa, ni, pa, ga, re, sa.
And this is a drone, so this is just playing that main note and not changing. We don't really have harmony,
Right.
but we have a flow of the melody. Can you sing that the way you sang it with the connection, the way you sang it to me last night?
Definitely. Yeah, so the drone is the first and the fifth. So it's sa, pa, sa. And that just helps the artist stay on their tonic that they've set. So what I meant when I said that the notes are sung in relation, there's a relationship that the notes have. There's a character and a movement that's expected. So somebody who's studied Indian classical music, if I say I'm singing Hansadwani, if I just sing sa, re, ga, pa, ni, pa, ga, re, sa, they'll say, oh yeah, those are the notes, but I'm still not hearing Hansadwani. So they would want me to focus on the second note, for example, is a point of emphasis. So sa, and the connection. So sa, re, ga, re, sa, ni, sa, re. So they'd be looking for phrases like that. Some emphasis on ga, the third note. Sa, re, ga, pa, ga, pa, re, sa, ni, sa, ga, pa, re, ga. So these are phrases that are expected, and different ragas can have the same notes, the exact same notes, but it's the way that the notes are sung in relation to each other, and that's sort of the glide, the connection between the notes, the phrases, the personality. So if there was another person here wearing a purple shirt and the same outfit as you, I would know you are you because of your character. So it's sort of similar with a raga. Even if the notes are the same, the appearance is the same. It's the character that makes it what it is.
And I will explain that this synthesizer, which you can see is called a continuum fingerboard, is named that because it does play continuously. So not just, but. So when I'm trying to imitate what Samita is singing, this instrument works really well because I can come up with a kind of a simulacrum of what you were singing. So that's how we selected this particular instrument to go along with the singing. Tell us just a little bit about this first song that we're going to do.
Yes, so I'm going to present two compositions in this scale, in this raga, in a very traditional manner. So that is that typically an artist, whether they're playing an instrument or singing, in North Indian classical music, you will sing a couple of compositions in the chosen scale. The first composition will be a very slow tempo, where you slowly develop the rag by singing different phrases that focus on the notes of the scale. And this is all improvised. That's an important point to make too. So,
Hence my terror.
I mean, yeah, when you don't know what you're going to sing, I mean, it's also exciting, right? Because
Terror is exciting.
because every time an artist performs a raga, it's going to be different because it's depending on how they're feeling that day, it's going to come out differently. But there's parameters, as I said. The notes are just the notes that exist in that raga, and you can't go out of that boundary. So basically, what I'm going to be doing is singing these two compositions, developing the raga. After I'm done singing the full first slow tempo composition, I'll sing a series of improvised phrases, and you can do that using the sound ah. That's typically how it's done. You can also use the words of the composition, and I will sing the whole composition with these improvised phrases, sort of building the rag. And then I'll conclude that first slow tempo piece and sing a second faster tempo piece.
And just very briefly, because I want to go all night, but we can't stay all night, talk about how the rhythm is. So time is a very inchoate, a very undefined thing, and yet music chops up time in certain ways. So how are we chopping up time in this kind of music?
Right, so the first slow tempo piece is a 7-beat cycle, but it's a very slow 7 beats. So if for those of you who are maybe familiar with rhythm, or if you've studied music, when you listen to the rhythm that's playing, if you count it 1 and 2 and 3 and 4 and 5 and 6 and 7 and 1. And so the riff falls on the first beat. So you'll hear me singing Me Hari. That Hari, that's something you can listen for. That has to land on the first beat.
Which is, for the Western ear, sort of unusual that a dissonant note, that's our main note, that's our drone, and that note right next door is landing on the strong beat. But it definitely gives a kind of a tang to this particular raga, I think, to my ear.
Right, yeah, and it's that second note, as I said, Re is an emphasis note in this rag. So the person who composed it, Me Hari, the fact that it lands on that note on the sum was something that was probably in the person's mind when they composed it, right?
We're going to play this one for you. Thank you.
Ah
Thank you so much. Wow. Gorgeous. Fantastic. All right, we have another much shorter one, and that'll be it. Tell us about it.
Yeah, so this is in Indian music. There are many different types of compositions. So the compositions I just sang are pure classical, but this next piece that we'll conclude with is a more devotional song. It was written. The poetry of it is written by a saint named Kabir, and the special thing about Kabir is that he was embraced by people of all faiths, all backgrounds, all cultures, because there was a universality in what he wrote in his poetry that sort of just brought everybody together.
So you're talking about Hindu religion, Muslim religion, no divisions. Everybody was drawn to this particular body of work.
This body of work, yeah, by the Saint Kabir.
I think that's a wonderful thing, especially in this day and age.
Yes, definitely. And so this scale, the raga that this piece is in, is, sorry, I just have to get this tuned properly. It's very different from the rag that we just presented. It has all of the notes of the scale. So the last rag was pentatonic. This has all of the 7 notes in it.
Just to clarify, as it sounds, a pentatonic scale is a 5-note scale, so it doesn't have, typically a major scale has 7 notes in it. So a pentatonic scale is a reduced number of notes. Right. By the way, what you're about to hear, this raga blew my mind when we were practicing it. It's an absolutely gorgeous collection of notes.
So yeah, this raga has, I'll sing it for you so you can get an idea of the feeling of it, the character of it. So it's sa re ga ma pa dha ni sa ni dha pa ma ga re sa.
What's so cool about that to me is the bottom of it. It's like our major scale, and then from here, it's like our minor scale. So it's major on the bottom, minor on the top. To my ear, that gives a really delicious and kind of exotic flavor to the scale. I'm not sure, how does it strike you when you hear that? Probably doesn't seem exotic because you've known it for a long time.
I definitely gravitate towards this melody. I find it really beautiful to explore, and also, it makes me feel a combination of, I don't know, it evokes like almost like a pensive feeling in me, like a very thoughtful feeling. But what's really wonderful is it's really up to the listener. With any piece of music, really, right? It's really up to the listener to decide what it means to them.
Talk to us afterwards, and you can let us know what it meant to you. This is our final song.
जपो भाई साधो अजपा जप जपो भाई साधो जपो भाई साधो अजपा जाप जपो भाई साधो अजपा जाप जपो भाई साधो सांसों की कर लो माला सांसों की कर लो माला जपा जाप जपो भाई साधो अजपा जाप जपो भाई साधो हाथ सुमरनी हाथ सुमरनी हाथ सुमरनी मगन कतरनी हाथ सुमरनी मगन कतरनी यह क्या रच दियो जाला यह क्या रच दियो जाला लोगों के भावे भक्ति कमावे लोगों के भावे भक्ति कमावे साहेब के मुख काला जपा जाप जपो भाई